controversial subjects with the facts can be tense, but we are a science here to make things make sense.
So it's the year of the rapid antigen test. Like maybe it is.
Yeah, it's interesting because obviously I've heard of it, but in the recent months it's become like so prominent.
Hot commodity.
And here we are yet again 2 years later talking about COVID. It is actually crazy that it's almost 2 years to the day we're coming up on. Like in a
March
month
22nd.
That it all began.
But don't you think it was
Never would have thought we'd still be talking about it, by the way.
The rapid antigen test was like the Tickle Me Elmo of this Christmas.
Oh, yeah.
It was like everyone couldn't get their hands on it.
Yeah,
Like,
it was like the toilet paper of the beginning of the pandemic.
Yeah, it was like the jingle all the way of, it was like, I need to get a rapid antigen test to go home. Like it was crazy. Yeah. And I relied on them when I was at home with my parents. So like we'll talk and stuff. Everyone in Ontario had a crazy Christmas time with COVID because Omicron came like just in time to really like throw a lot of people off. Like it was I thought it was a fascinating time like living in this city.
Because it obviously hit like obviously South Africa and UK like a little while ago before us, and then it just was like starting as we went into the holiday season here.
And it really is so much more contagious. It happens so fast.
Yeah, like the whole pandemic, so a year and 8 months, I've maybe known like one person who got COVID who's like a friend of a friend.
Yeah.
Or maybe a handful of like stories that I've heard, but like it suddenly became like so many people I knew personally who are like close friends were getting it. And people who have been like super careful the whole time, wear masks, are double vaccinated. So obviously it was like a big shift. Yeah. And now I think a lot of people in Ontario at least, but I think this is happening in America as well. In the UK, it's kind of like everyone now is suddenly knowing a lot of people who have got it.
It's so weird. Like this time last year, we didn't have vaccines. I was still so scared. Like if I had known someone who got it, I like would remember them and like think about them on my walks. Like I hope their lungs are okay. And now I'm just like, oh my God, like all my friends just got it this like one weekend and I randomly didn't. Like I was so lucky, like because it happened so fast that like I was out the weekend that people were like getting it in Toronto and not knowing.
Yeah.
But it truly is a different virus. It's a more contagious virus. And it is, at least now, like it is less it's like less physiologically damaging than past variants, and the people who are vaccinated really helps as well. So like there's a lot of reasons why this like caught so many people off guard. It's like not like,
well, and it's just so fast the way it happens.
It's crazy.
But so how do we want to structure this then?
Well, okay, my point of that was that it was the first time that I kind of did a little rapid antigen test in order to like, I tested negative PCR antigen. Then we got them and I did them at home with my mom.
You're right. I've only done a PCR until recently. Like I've done a bunch of tests, but they've always been PCR.
When we did the show, it was PCR.
Was it?
Yeah, it was PCR because rapid antigen are the ones that they would tell you right then. It was all going to a lab.
You're right. Like we wouldn't, because we would not be getting our results. It was more like when we did the show, if it had come back positive, obviously they would tell someone. But
it was PCR.
Yeah, you're right.
It was. Right. I'd never done an antigen test until this Omicron wave, and that's why this show is like. I think it's important we all understand how they work, what they are, because I do think it's going to be like the year
of the like rapid tests
of the Tickle Me Elmos in your house need to be antigen tests if you want to like feel okay with like certain things. It's crazy.
Yeah.
Okay.
So the big question then is like, what is the difference between the PCR tests and the rapid tests? Do we want to start there?
Yeah, do that. Go.
Oh, no, I thought that's what you were going to explain.
What?
Oh, what are you explaining?
I'm explaining how they work.
Okay, so you first went. Okay.
Yours is like right in front of you says differences in the two.
Oh, no, oh my God. See, you do read my computer screen. I hate that. You're always staring at it and I'm like, you promised me you weren't
I just did for that moment of panic.
No, because you're going to explain rapid, so I don't want to steal it from you.
No, just go.
Okay. Well, basically, so a rapid test is like looking for the antigen on the virus, right? Which protein, is just like the protein on the virus surface. So when you take it, if that gets detected, it comes back positive. Whereas the PCR, which stands for polymerase chain reaction, is looking for like a DNA sequence. And I actually didn't know fully how this works, but it's like, obviously when you have a tiny amount of DNA, like there's not much you can do with that, even if it's not to study like viruses. Like PCRs are used in lots of different scenarios as well. So they've come up with this technique where they use something called a primer. And that primer will recognize a very specific sequence of DNA. And that DNA, obviously, in this case, is specific to the virus we're looking for.
Yeah.
And if that primer is found, it'll start to basically amplify that DNA and make like
cool.
millions of copies of it.
Wow.
Which makes it suddenly way easier to tell that it exists.
there.
So instead of
Ooh, that's so
which is why
cool.
PCR is so accurate, because it's
Yeah.
like it can find this tiny bit,
piece.
and you're using like laboratory equipment. It's obviously like way more
Oh my god, it's so
sophisticated.
cool. And then it uses like cell bio to like replicate it.
To
so that
replicate
you,
it
like-
over and over and over, and so you're like in your sample, you're like, okay, clearly
God,
there
there's
is
so
COVID-19 here. And
Cool.
if it doesn't find that, it just doesn't do it. And so that's why PCR is so accurate, because it can go off of like such a small amount of DNA. That doesn't mean it's perfect, because it might, if there's very, very, very little in you, you might not find
Like,
it.
so little,
but because
yeah.
the virus is like, an RNA that, and then PCR like goes and turns it into DNA and then takes those strips and then starts duplicating it. But yeah, that's kind of like the ultimate difference. And so PCR takes longer typically because it has to go through more lab processing, whereas the rapid antigen is just like, is this protein in your nasal swab?
Okay, so now I'm gonna that's perfect. I'm just gonna go explain how the rapid ones work now, really intensely molecularly.
Okay, great.
Okay, because like that's so I didn't know that about PCR. That's so interesting. But I just knew it was more accurate and that it used DNA and not the protein. So like these rapid antigen tests, the ones that I've used, have you done one? Like done it yourself at home?
I've never self-administered one, no. But I have self-administered PCRs, like when we were in the UK.
Oh yeah.
But when I got my rapids done, they were done by like a physician.
Oh, okay, yeah, because I got to do one with my mom and it was fun. It was like, okay, we're scientists. Like it is like an at-home, like it's so interesting.
literally is,
like
yeah.
it's like a weird,
right.
like
What a good
everyone's
chance.
doing
Tell me
like
about science.
like, oh, I got a science kit for Christmas.
Yeah,
like it's an
seriously.
antigen test. Okay, so like one thing I loved about doing it is you put the little drop. You essentially take the little swab, put it in your nose. You do it like one nostril, like 6 seconds. You go deep, but it's like not that bad. Then you put it into the liquid. But when you take that liquid with your like sample in it and drop it in, the way it spreads on the little piece of white paper is so cool, because it's like
you
capillary
see
flow. It goes like
it
oh
yeah, I've never got to watch that.
and it's like actually like I was like, this is cool science. And so then they're cool. So that's called capillary flow, which I then was like, I became obsessed with that because that's a weird thing you learn about in like cell biology and you teach it as a science teacher. I remember capillary flow.
Yeah.
So it's like, what it is because water would more rather be attracted to itself and walls sometimes due to hydrogen bonds, like surface tension.
Okay.
Like water surface tension, so interesting that if you put a thin enough tube or cylinder into water, the water will actually like go up the tube a bit. You like you can sometimes picture us seeing that happen.
And I think this is also related to how trees like get water
Exactly
up through them, right?
Yeah, it is capillary flow is how like it goes up
one
the
of the
xylem.
tactics of
Yeah,
these,
it is. And then also the fact that it's evaporating out and like pulling it. That's so true. It's like the xylem is the tubes of the tree in the trunk, and due to the way water wants to bind to the sides of that xylem and to itself, it goes up the tree. So it's that exact, there's these little capillaries in that white paper in the antigen test that pulls the liquid along it.
Okay.
Which is like it's cool to watch.
Yeah.
Okay,
I
so
know. I'm sad that I've never got to like put the drop on.
I know next time you should be like, can I put the drop? Yeah. Be like, I'm a science communicator. I'd love to see the capillary flow.
I have a lot of experience in this field. I worked in a lab actually.
Some people call it liminal flow if you're talking academic. Okay, so I just like love that. And also surface tension of water is so freaking cool. And I started to remember, I'm going on a bit of a side note here, but it's still about the concept of capillary flow.
Well, good thing that's the name of our show.
Good thing we called this thing the Side Note Podcast. Controversial subjects and the facts can be tense. So it's like, remember when people said that if you jumped off a bridge and like threw...
Would you follow your best friend if they jumped off a bridge?
No,
Is that what we're talking about?
no, whoa. If Johnny jumped off a bridge, would you too?
That's kind of like, yeah.
No, wait, is
wait.
this
No, I'm
this
joking.
not a thing that like...
What are you asking?
Mythbusters did an episode on it. That if you jump from a bridge and you're falling from a bridge or something
Yeah.
and you don't want to die, you should throw like objects down to break the surface tension of the water and therefore you won't die.
Oh.
So that's not true. But you've not heard that?
But no, I've never heard that. And also, what objects where?
Okay, it's
you're
like-
falling
it's like-
from 100 meters and you're just like, okay, I guess I have a lint that I could throw.
Okay, the concept to me was like a worker. He's building a bridge and he's got all his tools. And oh no, the harness breaks and he's fallen, but he's got his tools and he whips down his wrenches, breaks the surface tension, falls in, doesn't die.
That can't be true. And you said it wasn't.
But okay, the physics of why people think that's true is what I think is so interesting about water. Like the surface tension of water, when there's enough like velocity from high enough up, the water surface actually acts like a solid almost.
Yeah.
And it's because water would rather be attached to itself than air. And so the more like a belly flop hurts more because when you're splayed out like that, you have to push more literal water out of the way because it needs to be pushed because it wants to be attached. Whereas when you do a dive, there's so much less water to move and you have more weight. That's why you don't like get that. It's like surface tension. It's cool.
Water
I don't know,
can
I love
really
water.
hurt. Yeah, I know. I think maybe this was a Mythbusters. Like, can you like break your legs landing in water? Right? Like if you
die. No, you die.
Oh, you die.
You literally die. You break your legs for sure.
So it's like cement. It's just like
It's
falling on the
literally
ground.
like it's like hitting.
I feel
a
really
solid surface.
unwell right now.
Okay, anyways, that's like why people die when they fall. Capillary flow is related to surface tension. Surface
Come
tension
back
is so
to
cool.
capillary flow. Okay, yeah, because I felt like that went to a dark place.
No, yeah, it's just I don't know. I've always loved that fact. The fact you didn't know it makes me feel like I'm like a murderous freak. Okay, so then there's these two little spots where you're looking for a red line. And it's T and C.
I was like, in the water to aim at?
That's the blood. Okay. You're dead.
Back to the rapid test.
Yeah, the rapid test. You're looking for these two red lines. One's T, one's C. At first, I thought, confusing. Why wouldn't it say like
what does T
positive,
and C mean?
negative? So now I'm going to explain what it means.
Okay, because I truly have never seen one.
T, the red line at the bottom means test. Okay. And C, the red line on top, means control. Okay. I can't believe you haven't seen this. There's so many memes that are like how to get out of having to see your family.
Sorry,
and then,
I have seen that. I know what they look like. I just never realized. Like I haven't stared at it and been like, what's T and what's C?
Okay. I just like if you see the memes, it's like there's always two lines when they're like how to stay home from how to not see your family on Christmas. Like, oops, I got it. But they're drawing like a red line.
Yeah.
So the two lines is the means that you have it.
Okay.
So when you put your literal liquid from your nose into the little dish, which everyone's probably done if they've done it themselves, and it starts to capillary flow across, the first thing that liquid does is it goes over these series of antibodies that are set up right to the right of where you drop the liquid.
Okay.
So those are like these primary antibodies, and they are looking for the protein.
Antigen.
Yeah, they're antibodies for the antigen of the protein of the virus. Also, when you put your swab in that little liquid, it actually breaks it up and therefore allows the protein to be more easily accessible. Yeah.
Okay.
So then it goes. So if the protein is there, if you are positive for, let's just say positive for COVID-19, and the protein is there, those first antibodies will bind to the protein and start to move across.
Okay.
Then the T section is made of proteins that also bind the virus protein, but on a different, like it literally on the other side.
Yeah.
So therefore, that antibody attached to the viral proteins moving along, it binds to it and it kind of like literally physically moves it. And then it creates this dye that makes the red line. Then it keeps on going. And there's this other set of antibodies, which is where the C is written. Yeah. And that is just meant to bind to the antibodies on a different part at the very beginning. So that one, no matter what, should
go off every time. And so if that doesn't go off, it's maybe like your thing is faulty.
It means that the antibodies were faulty like at the factory. It's like you can't rely on that test because there's really complicated 3 parts of like that strip. Very like scientific the antibodies need to be properly like put there. Yeah. If not, like, and then so sometimes people will get,
or maybe like manufacturing, maybe they just like didn't spray it that day.
And so if you get just the T and no C, which means control, it's a debunked test. You can't trust it. So it was essentially like
debunked.
Debunked. But when you think about it, you're like, oh my God, it's literally just taking my virus, attaching it to an antibody, flowing it through capillary flow on this weird piece of like science paper. It's either going to click, like literally hit the antibody, turn it red at T, or it's going to not have it have some other like not this specific antibody like disease. Maybe it has like some other cold virus, so it doesn't hit it, but it does because it's an antibody bound to something, hit the other one, the control. So we now know it's like got there, and then you decide if you had the protein or not. It's cool.
Amazing.
I didn't think it would be little things like that small in it doing 3 things.
I never thought about it, had no idea. And sometimes I just get overwhelmed by how smart people are who figure these things out. Like I know to some people it's actually probably really simple. But to like get to
no, it's not simple.
I know it's not. And just to get to the point where even realizing that there's just like scientists and genius microbiologists around the world who like understand how to like get these antigens and antibodies. Like not only do we like look small enough, we developed microscopes to learn what they were. Then we were like, how can we actually use those, like mass produce them in a way that allows us to detect? Like it is all genius.
Actually fascinating. And it's exactly why people need to not be so down about like, I mean, sorry, everyone can be down about COVID, but it's like interesting is it's like we're in a different stage and science has been so helpful. Like this time last year, these tests were A, being invented, B, were probably like barely being used because there weren't factories and enough science to make them.
Like they might have been, I don't know if they were invented.
There was probably just frantic scientists being like,
well, even
smoking
like the
a cigarette,
PCR
being like, I'm almost
used...
done my design.
Yeah.
You know what I mean? Like the design's almost done. We're almost getting there, folks. And it's like, now we got there.
It's crazy.
It's so cool.
Unbelievable.
And it is why many people, now we should go on like, how do you use them? Because it is now why I think a lot of people are using them to be like, do I see my family? Which is a morbid thought. We're back to COVID being hard.
Yeah. Well, one thing I wanted to mention about antigen antibody, because this question popped up for me, and then I like obviously like had to figure like knew the answer sort of thing. But I remember wondering like, why do viruses have antigens? Like, doesn't that give them away?
Yeah.
But it actually is just that antibodies, whatever they like, this is your immune system trying to find things. Like the antigen is just whatever protein your antibodies are connecting to.
Yeah.
It's not like because you know when you see science drawings, it's like a Y that's like coming
so
at me. And then,
it's so abstract. The way people talk about antibodies is so abstract, and they like clip on the thing.
Yeah,
that's exactly what they look like.
And
in
like,
these diagrams.
why does the virus have those then? Just get rid of them. But it's like actually just a representation of the proteins that make up. Yeah. And those proteins on the virus could be used to help them get in the cell. Could be used to like help build their structure. Like they're important parts of the virus. The virus can't just like get
antibodies evolved in direct like combat of viruses. So it's like evolution like molding, designing into each other kind of. Because that's what your body's designed to fight, because it's like the small things that kill humans. It's so freaking cool. It's so funny because I was reading about all of this on like physics, like antibody, like
deep web forums.
No, literally like Nature magazine. Just like a complicated article. And it's so funny when they show the visual diagrams, like they've gotten more complex and like more artful than when we were growing up, but it's still that weird Y thing with two little lines that like literally hit the
and then the other one's a little like, mouth that like comes and
I'm like, is it a mouth? And it's like, it's not. Like it's just like a visual representation for us all. It's just like funny because I'm like, I thought it would have advanced to a new like more clear thing. But
well, I think that is that part of science when you realize to teach things, you sometimes have to actually teach it wrong first to make it simple. Like a lot of science things you learn in grade school are kind of debunked and restructured in high school, and then in university they're actually like, well, actually just forget all of that because it's not true. And I think that and that's a lot of visual representation is just so people can understand what it's like, because when you see chemical structures under, like when they actually draw them out in these different 3D ways, it's like
so complicated.
Okay.
Like I'm not gonna do a blobbity blob with
and
like
there's no intuitive
so many like weird liquid sides.
Whoever came up with the analogy and started using it in textbooks is just really smart, where they're like, it's like a key and a lock. And so like, let's just draw it like that.
Ligand substrate vibes. Okay. But know what is so freaking cool about that is that they actually now can know the shape of proteins and molecules. And it's, of course, they're not the little.
Still is a bit of an abstraction, isn't it?
Oh, well, no, it's not now. Well,
like when you look at PyMol, for example, that software, like that's what
it looks like in 3D physical space.
That's what I wonder.
That's so cool. It is an abstraction with color, and sometimes in order
freaks me out.
In order to understand the liquid shape of it, they like use a software that like uses essentially like arrows and these like tube screws to represent specific folds in proteins. So there's versions where you see the physical shape where it's represented by like symbols they use to represent how amino acids fold up. But then you can press a button and it becomes the like 3D Pixar Toy Story version of the shape of it, and that is real. That's the shape of it.
I need to look at more of those. To like
those are cool. But the thing is, they're so complicated you would never draw them. Like they're in our diagrams. They're a circle and a triangle.
And also, I mean, like I don't know. I am curious how literal those shapes are in terms of. Obviously, like when you get smaller and smaller and smaller, when you see people trying to express like what electron orbits look like and stuff, you start to realize like it is often an abstraction and like.
But the thing is that the weird thing about electron orbits is that that's actually like closer to like quantum.
Yeah, and it's
physics.
like probabilities about where it should
that's
be as
so
well.
weird. Whereas these proteins are so
physical.
big that it is a physical.
That's
system.
fair, I guess. Like, but you're saying it's big, but also this protein is like a piece of a virus, right?
Yeah, no,
so like it
so
is
small.
It is
Sorry,
actually
it's
tiny.
so small. So what they do is they shoot lasers at them. I feel like I've talked to you about this before.
Yeah.
They shoot lasers literally at these tiny, tiny proteins. And based on how the light scatters, they're able to see.
It's so messed up.
I know, it's crazy.
And
They're
I'm
able to
like,
put it
well,
back together with computers.
how did they get it there? How were they like, well, let me just put this
Well,
little
that's when like
thing
you're
there.
like you hang out with the professors and they're literally like they're socially awkward because they're so smart. Like, you know what I mean? The people who like do that, they're like, well, you see, and they're like always just like on their computers. Like you can't talk to them because they're like always coding, and you're like, well, I'm so happy that you're so passionate about this because without you, we would not understand how to make medication. Like,
It's so
thank
true.
you.
Yeah. There is a level of science that I and especially like the technical part of science that it's like, thank God there are people who
Yeah.
like that. Like I love the knowledge it brings me from like the bigger context and
Yeah.
even taking biochemistry. Like I loved it. But to do the technical work of figuring out what that was is such a different thing than
Oh my
learning about
God.
what that thing is. Do
And
you know what I mean?
Yeah, for sure. Like they're called like
It's amazing.
world-renowned scientists, and they exist, and they're truly the most fascinating people in the world.
Yeah.
Yeah, no, it's true.
Because
It's
you
like,
have to have such an interesting personality to just
Yeah.
like, I mean,
or
there are
just
lots of
literally-
brilliant-minded people.
I think they're just so smart that they can't not.
But I mean, to be honest and to be fair, I know some people who do work like that are completely, totally social and amazing.
Oh,
Like,
I'm not.
I know we're playing
I'm
a
not
character.
saying you're not social. Sorry, I made a voice earlier that was like.
Well, I'm
I'm
thinking-
talking about intense intelligence and work ethic. That is
Yeah,
like
on the
unfathomable.
people who are like on the cusp of
They're so
like
smart.
breakthroughs, because there are lots of technicians, and I will say I was a technician in a lab, and so like there's lots of levels, right? Like I was doing
Yeah.
like different ELISA testing and stuff, and like understood the basics of how that worked. I didn't invent it though, but I was like
That's what I
somebody,
mean.
and I
The
think
people who invent it are the ones who are just
they
too
are,
smart.
but I guess sometimes I'm like, science is so wide that there's so many people involved, and it's not like someone's
Yeah,
like,
that's true.
Eureka! I figured it out. It's like slow
Yeah.
reasons that
That
we
is
figure-
like such science journalism to be like, oh, whoops, like the penicillin, like threw out the penicillin in the mold one is
Yeah.
like so epic. That's a eureka moment, I'm sure. But it's like, is that even freaking true? Because it's always so.
I think
you're
it is, but
hyperbolic. There's so much
it's
intense
rarer
data that's
and
first.
rarer, I think, for that to happen
Yeah.
because we're in such a specialized world now.
Yeah.
And like back then, humans were stupid and didn't know anything. I
Mm-hmm.
mean, I'm sure people in the future will say this about us, but like it was easier to have like a very novel, like visible discovery.
Yeah, that's
At this
true.
point, it's like we've seen it all. We need to have
Like-
tiny ass microscopes and huge fucking telescopes to see anything new
Yeah,
now.
the first time they like was a microscope and they were like, there's things smaller like in our skin. You know what I mean? Like that was an easy discovery. Like they were like,
Yeah,
there's
they, like,
literally moving
Put
things.
the freaking goggle on, and we're
Yeah.
just like, whoa.
Yeah. So yeah. Anyways.
Going back to rapid tests, I recently saw a study came out. It was only 30 people, but it was people who were likely exposed. And they found that PCR tests could catch COVID-19 3 days before rapid tests. This paper has not yet been peer reviewed. It's like really new.
Yeah.
But they were trying to figure out like, okay, what is actually happening? Like what tests are the most useful? How can we accurately use rapid testing? And so it was just like interesting to know, like PCR is way more sensitive for that reason that I said. It can find a tiny fragment and then just like duplicates it and replicates it with also complex machinery that's like different than this tiny little plastic device you can use.
I think people just need to understand how they work in order to understand how to use them and not, you know what I mean? It's not just like, people always want a black and white, like, do they work? Do they not? It's like, it's all based on science that has a sliding scale of meeting the needs that you need to know for your point in that part
and
of the
even
pandemic.
That's like a really good way to put it, even from a societal level. It's like, what is the needs we have right now? Like, do we need PCR? Yes, better, but they're kind of like rapid testing so much more accessible. So say PCR is 99% and I think they say rapid's like 80 to 90%. It's like, that's still really good. And if it's way easier and people can do it themselves, like it's a really great tool to have with a couple little caveats. Like you'll get more false negatives and
more false negatives.
Yeah.
So,
So you could have a negative and still be positive. And knowing that like it's more likely to catch it when your symptoms are high
And
because there's lots of virus in your nose.
And knowing it didn't like do some crazy, incredible cellular biology to PCR it into thousands of the virus for someone to see it. Like it wasn't one of those.
That's
but
why.
it just like found an antibody and went on a little piece of paper.
So this is what they say about the rapid tests is that if you get a positive on them, it's like you probably had COVID.
But there is such thing as false positives too.
Okay, there is, but
it's rare.
In these tests, like there's something called specificity versus sensitivity. So specificity is like the rate of false positive.
Yeah.
And sensitivity is like the rate of false negatives. Okay. So PCR is like great at both of them. So if you have a positive, you're almost certainly positive. And if you have a negative,
it's specific and sensitive.
Yeah. Whereas the rapid tests have a lower sensitivity. So that means they bring back more false negatives.
Okay.
Does that make sense?
Yeah. So there's more, you're more likely to have some of the protein, but when you do it, it's not as sensitive and doesn't like bind to the antibody, and
you may not find it. Yeah,
there's not enough. There's maybe not enough, and so you don't get the like.
You get a negative result, and you think you don't have it,
and then you go do something, and then you go, oh fuck,
I actually was sick, and I think a lot of people are experiencing that. People are like, I'm sick, and yeah, you might not have COVID, but you might, and if you only had a rapid test, it may not have like gotten the antigen.
That's why like the one consensus I could find about what they're great for is for if you have symptoms and you say need to go, like you are in an emergency and need to go somewhere. If you could do it, you could find out, oh, those symptoms aren't COVID, I can go. It's like, because if you are like sick and you have something in you and you're concerned about that, and you put it like on,
because it
like
could be a different cold that you're way less worried about than
whereas when you have COVID and if you have symptoms, you will definitely get it. Like if you have symptoms, there will be enough of the virus likely for a antigen test to work.
Yeah,
for the specificity to work because the
yeah.
proteins are there if you're feeling symptoms from COVID. So if you have symptoms, you know what I mean? Like, and like it's a good time to find out if what you're sick from literally that has like snottiness, whether it's COVID or not,
or some other thing bothering your body.
But it's also so funny because it's like, and then you just go maybe give your grandma that other thing you got. It's like if you're like
exactly, it's like weird that sick, we're like it's made me reevaluate. Like when we're sick, we probably just like shouldn't be like, well, I'll test and just make sure it's a different virus.
Yeah, because like okay, like literally that's what we did with my family. We all tested for COVID, and my little nephew and my sister like they tested him because he like had symptoms, and then the day of we're like, yes, oh my god, we're all negative. And he came over, he was so sick, like I've never seen. He was like, we were like, yay, dinner, and he was
tired.
He literally was like, I'm just gonna go lie down, and he like went and lied down, like couldn't eat because he was like so barfy. But we were all just like, yay, no COVID, no COVID. Then at the end we were like, well, we're definitely got like whatever that was, you know what I mean? Like we're like, sure, no COVID, but we're definitely all gonna like puke tomorrow. Like he was like green in the face, but we were like, yay, he's here.
Yeah, what a funny thing.
Yeah,
I hope when people leave the pandemic, they are like, if I have symptoms, I just shouldn't go.
Yeah.
If I'm like, because the amount of times even I've done this where I have a cold, I'm like, oh, it's just a cold, and I'm still like hugging friends.
I'll be like, hey, like I used to like love to party. That like if it was like a really good friend's birthday, and I was like, hey guys, no, don't worry, I'll just drink through it. Like, do you want to share cuts? And I would just like get drunk, and I remember thinking like waking up so hungover and being like, I feel like the cold's gone. Like, cause like you're hungover, so you're painful in a different way. Like my head hurts, but
like it cured the hangover.
I'm literally like alcohol cures. Like back when I was literally so young and dumb.
Okay, coming back to testing, I have a little unique story that I'm going to share, which is so talking about specificity and sensitivity. Before visiting some friends, we were like, let's just get rapid tests. My rapid test came back positive.
Oh yeah.
But
okay, he's going like, you're going into your. Let me check COVID.
Well, I don't know. Okay, I had no symptoms.
True, I had no symptoms.
I felt fine. Was like ready to see people that day. So obviously didn't.
Oh yeah.
Less than 24 hours later, got a PCR, was negative.
So Mitch had COVID for 2 days, and it was so weird.
Well, like for 10 hours.
Isolated from you. But then he was like, so Mitch is so funny because he's always
I'm always sick.
You always are sick.
No,
like,
I'm not actually sick. I always feel
every day he's
shitty.
like, oh, me head. And then I'm like, he's like, oh, I don't know, Greg. I think I'm sick. And I'm like, okay. And then he's like, oh, maybe I need coffee. Then he like has a coffee. And he's like, oh, it was a coffee. I was like, yeah, it's called like getting up. Like, yeah, it's coffee. So like, you're always kind of like that. So then he's like, he gets his positive test because we're randomly gonna go away, and it's like, okay, we now have to isolate. I'm like, oh my God. And I'm looking at you. I'm like scared. Like, is it coming for me? Like you have,
Yeah.
and you had also just gone home and seen like your family's bigger than mine.
Yeah,
So
yeah,
like,
yeah.
whatever. We were just like, oh, you could have gotten it. Makes sense. So I isolate from him. And like every text from Mitch is like, feeling best I've ever felt. It's like, it's so funny. He's like, woke up, didn't need coffee, best day ever. And I was like, I'm pretty sure you're supposed to like, you know, if it's like an antigen test, you should be having COVID.
Crazy.
You should be like, and then you were like the best you ever felt. And then that made you, because New Year's was coming, you're like, I want to like, obviously want to see each other. I was like, you got a PCR test to be more accurate. It's negative. So it's like, did you get a false positive?
Well, that
was you boosted and therefore, you know, you just got rid
That's
of
what makes me confused about this, knowing that PCRs. Okay. And also, and like, this is not a drag of healthcare or anything, but when I was there, it was like there were so many tests that have just been done. I'm like, did I get someone else's result?
That was private. That was a private company. Where
it was, but yeah,
that was the healthcare system.
Sorry, but I just was like, I don't want this
not to drag that private company.
Yeah, and I'm not trying. Like they were doing a great job, and they're probably so overwhelmed.
Oh, you think maybe it was mixed up?
Like, I don't think that is likely, but I just, when I got it, the like practitioner, the nurse or doctor, whomever was giving it to me, like he had like 17 other people's tests beside him, and he was just like doing all these rapid tests and putting them to the side. So it was like, he easily could have like wrote the wrong barcode on mine.
I love that.
And he had an Excel sheet open.
Yeah,
and he was like
that's an
typing my
option
name
too.
in, and I
That's
was like,
an
did
option.
mine get? But that just also takes more like, I would have had to happen to get a person who tested positive and then got it sent to me.
Yeah.
But it's just really weird given that PCR should be able to catch it both before and after the like window for the rapid test.
So you got a negative PCR. Okay.
Yeah, less than 24 hours later.
Yeah.
And
you never had any symptoms, and you felt the best you've ever felt.
Yeah, so that's why I was like,
and I never got sick too. I was like, if you tested positive, we had been.
Yeah.
I was like, I thought I would, and I never got, and I never tested positive because I also tested.
Yeah. So very weird.
So weird.
And that made me question whether or not, and this is like the last thing we can maybe talk about, like whether now people are like, Omicron, is it different? And I know there's...
Oh, is that sensitive to Omicron?
Is the test is sensitive? Yeah. And they are saying that they think early research shows that the rapid test may not be as accurate with Omicron compared to Delta.
It makes sense. And if you think about
It doesn't answer my question of why I got a positive.
No, but you can still get false positives. It could have been that. It's like, whatever. Symptoms are a really important part of us all understanding this too. You didn't get them. So like, you either did or you didn't get COVID. Either way, you're boosted. Like,
and I am triple vax. So maybe it is just like, I might have had the most mild symptoms. And I know I said this to you, and like, my dad was telling me, and I had to say my dad because I don't watch sports, but he was like, yeah, all these hockey players, because they have to test all the time, are all testing positive, but like 8 out of 10 of them would have never known. Like there are lots of people who are like, obviously all these hockey players are probably triple vaccinated as well. Like they have the whatever,
but also
access
probably like
to it all.
Spreading it because they do so much in a day.
Yeah, it makes sense that they're all around each other, but
like
it's
we're all at home and
interesting
they're
that
like
so many of them wouldn't have known in the same way. Like if I didn't test that day, I would never ever think I had got COVID, and even now I'm like questioning it.
It's so interesting about like the way that these tests are made on like a molecular level. It's like they would have to update them for a changing virus. Like they're building antibodies. We know now it's just antibodies glued to a thing. If those antibodies have been designed for Delta, then as they're gonna have to also change them. I bet they'll get more accurate. Specificity will go up in the next months, and it'll be even better.
That's what a lot of these like papers, studies are saying. Is like it just has to be updated. It's good to know what they're finding is they think that its sensitivity is a little less. It doesn't mean it doesn't work. It's just that means it's maybe a little more likely to give you a false negative than it was before.
It's so interesting.
And, sorry, I'm just going to say the tactic and the advice that they've been giving is like it is worth testing multiple times. It decreases your odds of a false negative, and it's always worth remembering that a rapid is kind of like an in the moment time, because it's unlike a PCR that can really amplify anything in you. You're really looking at like, what am I in this moment in terms of like, is it actually very present? And so doing it like I don't know what the actual timing advice is, like if it's once a day or every couple of days, just you're going to have a much better sense of whether or not you have it.
Also, it's like, this is such an opportunity for like governments to just like, let's just think of ours. It's like, send everyone rapid tests every 2 days. It's like really not that complicated. Like we all can benefit from them. Maybe as a government, like invest in your own freaking factories and people to make these rapid tests. Like sometimes I'm like, why are governments and people like so crazy? Also, it's like, this is a freaking like science, not versus. I hate saying like the giving like a moral conscience to a virus, but it's like it is like a human science versus like a microscopic thing that has killed humans for so long because of evolution. Like there's going to be ebbs and flows of like combating the thing.
Yeah,
and it's like
going to be. That's like literally, what do you call that? Oh God, I can't think of the phrase when like animals
kind of
are
evolution.
like, yeah, but when they're in a like chain of,
it's
like,
not symbiosis. Maybe it is. It's trying to kill us off. And
I feel embarrassed. I just can't get the word to my head. An arms race. Thank you.
Okay, well, what? This arms race? That's so funny.
Like
I thought you were gonna be like. I thought you were gonna be like they're like mycological homeostatic tension.
No,
the arms race.
An evolutionary arms race is when like species. So that's why some species are so specific and have really weird things because they've like evolved so closely with another competitor that they are like have extreme things in one direction.
Like it's so hard when people don't get vaccines. You know what I mean? It's why it's so hard when people like create all these like either unnuanced or so crazy nuanced explainers for like why we're doing this. It's like no, it's just a virus that has killed humans for a long time, and now we have science to combat it, and it's just trying. You know what I mean? Like it's just like it's not that deep. Like it's become this crazy thing. It's hard. It's become this crazy thing, but when you understand the science, you're like it's not that deep. Yeah, it's just like it's so simple.
Figure it out.
Yeah, we're trying to like not like, you know what I mean? Like anyways, it's weird because we now live in a really weird time with science.
We do, but it is amazing to think about, and it like gives me hope. And you know, most vast majority of people, not only in this province and country, like in the world, I think are like doing what they can and want to deal and want to support and are supportive of scientists who
and it's like
make technologies like this and are grateful.
And then
for
you
it.
know what? Maybe it's a little embarrassing when all the rich ass countries fucking hoard everything. It's like, oh, it's been 2 years and we've been watching what's happening. It's like, it's psycho. Like it's kind of
well.
psycho.
It also goes back to like
these people are evil, and we live in a country where
I remember like a year in, it was announced that Canada was like opening this like huge vaccine facility. But everyone at the time was like, well, it's too late. America's already got all the vaccines. Now we've bought all their stuff. Like the idea was like, oh, it's over. But you realize like how much money has to be put in up front. Like even I felt that at the time. I was like, well, like it's a little late to make this manufacturing center when we like.
But now
really
it's like maybe not. We need like next
well,
year.
you don't, and you don't exactly. You don't know when you're going to need it, and that's the whole point of investing in these things, like putting money into science and research. If you're only reactive, if you're only putting money when you need a solution, like you won't be ready. And that it's like, no, we do need a vaccine manufacturing plant, probably for the next 5 years, probably for the next
forever now.
Forever.
Yeah,
like I'm just saying, in 10 years,
it
might be literally a different pandemic. Like it's gonna be a worthwhile investment to pay for the science up front so you don't have to like pay the consequence and panic last minute. That is my PSA.
Yeah, no, it's true. Fun
Support
science.
science.
Hashtag fun science.
Hope y'all, if you make it to this part of this podcast and do not support science, wow. I actually am impressed.
Yeah, no, that is crazy. Like, that would be so interesting.
Like if someone's literally like, yeah, I made it to like 50 minutes in.
But know what I hate?
Disagree with everything they say. But like if you do, honestly message us. I'd be curious who you are. Okay.
Well, yeah, no, we're good. We're locked up, so we're not doing any tests because we haven't seen anyone.
Yeah, I know we probably won't do a test for a while now.
Maybe we will do tests soon when we open up.
Okay, well, stay safe, everybody. If you live in a country where Omicron's not there yet, brace yourself.
Oh my God. Okay. Peace.